study. We keep changing the names, but basically what we're doing is we are testing prescribed fire and prescribed mowing in different seasons, applying that to the landscape and then tracking what happens to the plant community in a nutshell. So that's, I haven't done this in a while. That's the basic question. So like, who would be interested in this kind of information? So when we started, this was really a question that ranchers were interested in as a land management study. So like, they're interested in keeping brush out of their pastures and increasing forage. And so it's geared to answer that question. I mean, is mowing similar to burning or is it not, and what happens? And so that's kind of how it was set up, but then it can also give us some information indirectly on other questions because we're tracking the composition of the community, so we know how it's changing. And so we can sort of obliquely get into some of these other questions. We can't directly answer them. And the other one that has come up more recently is wildfire mitigation. The fire department is very interested in this research, particularly with brush management. And so there are a lot of different types of questions we can get at with this, but it's based we're just tracking what happens to the plants. But you can kind of infer things from. So before we started, we knew a couple of things. So one of the things that we're doing is we are looking at fires, and we will default to talking about the fire more than the mowing just because we find it more fun and exciting. But all of these, we're mowing and we're burning, and we also have controls where we're not doing anything at all. But some of the things we knew going in, most prescribed fires happen historically in the wintertime. That's when all the prescriptions are for, but then our natural fires probably were happening more in the summer and the fall. And so we wanted to find out if doing that in a different season would change what happens to the plants. And so going in, we knew that dormant season fire, winter fire, sort of favors the big C4. So C4 grasses there's two different photosynthetic pathways. Most plants are C3, it's very efficient, but when it's hot and dry, but it requires that they have their stomata open so they can exchange, think back to your 8th grade, so that they can let gases in and out. So when it's really hot and dry, they dry out when that happens. And so the C4 pathway is an adaptation to very hot conditions. And so basically they separate part of the photosynthetic reaction so that they can keep hold of their water during the day. There's a third one that I won't go into. But anyway, those grasses, which are most of our native grasses, have this pathway. Burning them in the wintertime when they're dormant encourages them. So we kind of knew that going in. Growing season fire it knocks back their dominance just a little bit. And we want them to still be mostly dominant, but we also want all the other things, the forbs. Forbs are flowers. Anything is like an herbaceous plant that is not a grass or a sedge. At the end of the day, we want diversity. So we want lots of grasses, but we also want lots of forbs. And so we're trying to figure out how to get the most plants possible. And so we know that the growing season fire allows other things to come in. And then we also know that individual plants, it really is species by species. So they react differently depending on where you hit them in their cycle. And so we are trying to track that. How does this species respond when you hit it with a summer fire repeatedly? How does this one respond when you hit it with a winter fire repeatedly? Because what's coming out of this research and others is that one fire is very different than multiple fires over time. Things keep changing. And so that's the other piece. We're doing this study for a long time because it is really hard to sort the noise from the signal, especially in ecology. There's just a lot of variables you can't control or even know about. And so we need to do these studies over a very long time to start kind of picking that out. And one of the big reasons is that there's going to be a big effect, a year effect, on the research that may interact with your treatment effect. But what's going on? Hi. What's going on with the rain, with the temperature, with the world at large is going to move these communities around as much as or more than our treatments. And then to complicate that, they interact. So we are trying to pull out that. We're trying to, I sort of mentioned this before, we're trying to kind of predict individual responses on how a little bluestem will respond to these different treatments. how a big blue stem will respond. And we want to do that because land managers, it's very helpful when you're trying to plan management of your property. If you know, okay, I'm trying to encourage this set of species, or I'm trying to discourage this set, it's nice if you know what's going to happen. And so we're trying to kind of dial that in for them, whatever reason they're managing for. So this is the layout of the study. So to orient you, we are in this building right here. There's La Crosse, there's the parking lot, and so the study is all south of us. So we have several treatments. So this is simplified to just burn. So these are the plots. The plots are about 1.5 acres, give or take. And they're all replicated, they're all copied. So each treatment has 6 copies. So the treatments are, for the fire, we have summer fire, we have winter fire, and we have fall fire. And so those are these red ones. That's all the fire treatments. And then we have these yellow ones are mow treatments. And so the same 3 seasons, summer, winter, and fall. And then the mow treatments have an extra one, which is frequent mow. So that frequent mow happens every year. Whereas these ones, the rest of them only happen once every 3 to 5 years. And then all the green ones are controls. So we don't do anything to them except for monitor them and just see what happens, because we need something to compare it to. And then complicating this further, we've recently, so all of the treatments have 6. We originally had 12 controls. We've recently taken 6 of the controls and we're doing what we're calling a restoration treatment. So in science, you try to only change one thing, but these restoration treatments, we wanted something that we could demonstrate what an actual restoration would look like, which means we're changing multiple things. When you're doing land management, you never just do one thing. So the restoration treatments, we put them under a fire treatment and we're adding species. We don't add species to any of the others. And so we're monitoring that, but it's not really part of the study anymore because we're changing too many things, but we still want to be able to see that. So when you're on a plot and you see restoration, that's what that is. It used to be a control, but now we're adding species as well as burning it. Okay, so hi. There should be. So a lot of things can happen, and we can track them. So the basic variable that we're looking at is percent cover. So that's how much of the ground a given species shades, and that's what you'll be estimating. And so in the most simple situation, you have a plot with fire, a plot without fire, and in one of them, you have your cover there, and one you have your cover there. So great, I found an effect. That's cool. And then you have another plant, and you see this pattern. So there's no difference between them. This plant doesn't care. Great, if that was all that simple. Or you can get, this is what I call about a year effect. So you have a cover here in this year, and you have a cover here in that year. And we call it the year effect, so we don't exactly know what it's something to do with the climate, but we don't know exactly what's making it different. But it wasn't our treatment. It's the same for fire or no fire. It was the year that made that coverage different. And then it started getting more complicated. So you can see this one's having a treatment effect and a year effect, but they're not interacting. And this is where it gets really exciting, and this is Sean's life, is starting to figure out these interactions. So here you're seeing you're starting to see interactions between the treatment and the year. Y'all don't have to sort any of that out. That's Sean's job, but basically we're trying to sort these guys out, individual plants, into how they react. So you've got the insensitive plants that don't care what's going on with the year, they don't care what's going on with the treatment, they're just sort of always there in the background, and you will learn these first because they are in every plot, kind of at the same, most of these little small forbs, they're out there all the time, don't care what you do. There are some that will either respond to the year or the treatment, and so we want to know that. There's some that respond to both the year and the treatment, and then there's some that respond to both, but the way that they respond changes. The way they respond to the treatment changes with the year. And so these are from our first few years of data. We're going to make new ones soon. So this is Evolvulus cerisius. It's a little tiny forb, it's adorable. It doesn't care. It looks like there's no pattern because there's no pattern. It's just always there. It's always there in the background. This is one of our, this is Hilaria, it's curly tea, is the name of this guy. And so this one has a pretty strong summer burn response. So it likes summer burn. You can see how it's going up and it's not having much of a year effect. This is winter grass, so one of our few native cool season C3 grasses. It's also very, very resistant to grazing, so it's really common right now. It's having a very strong year effect, so that 2003, you see a big bump in it, but not so much a treatment effect. And then here we start getting the interaction. So this is Hall's panicum, it's another grass. So we're seeing these big, so we're seeing a year effect and a treatment effect. And then here is we're getting more complicated. So we're seeing more of an interaction. These are the ones that are the most difficult to sort of tease apart what's happening. But it's important to remember that year 2003. So it's not just our treatment. So 2003 was like drought year. As you remember, you saw winter grass spike that year. So part of this is due to interactions between the plants, not so much just what's happening with the seasons or what we're doing. So winter grass, when other things decrease, winter grass goes up. I just see winter grass. So yeah, what I said. Okay, so this is the more fun slides. So what we're trying to track is the underlying changes under the community. And so this is nice and visual because it's a big flower. And so over here we have Black-Eyed Susan. Over there it's Indian Blanket. Indian Blanket is a really strong competitor, but it doesn't love winter fire. It doesn't love summer fire. Black-Eyed Susan is a reasonable competitor, but not, it tends to get outcompeted by this one. But they differ in their response to fire. So when you remove when you knock down the population of this guy can pop up. And what we're trying to do in ecology generally is try to make, if we're doing land management for health, for ecosystem health, we're trying to make those patches. We want a lot of individual diversity within a community, but we also want diversity between communities. And so we want these little patchinesses to happen. And this is happening sort of across the board. It's just not as visual with these as it is with these flowers. So these were taken on the same day, and then the two plots are not very different, very far apart. The plots are all very similar by design, and so the only difference here is the treatment, which is fun. We have to do long-term research to figure it out. So back to our treatments. This is when we meet in the volunteer room, we'll meet there, and then we're going to head out to these plots. And so it's all on property, but it is it's a field day, so be prepared for that. This is just for fun. So this is one of the fire treatments. That's what it looks like. I like to talk about these nice straight burn lines because we're not just throwing fire around. There's usually questions about fire and why we do it, how we do it. And we say prescribed fire. Please don't say controlled burn to anyone who does this. It's like nails on the chalkboard. It is a prescribed fire. We have prescription. And so what that means is we're doing it under a very specific set of conditions: weather, humidity, the wind direction and speed is very important. And when we're applying it is important. And how we're applying it is important. And so just want to watch them wrap up. We are putting these where we want them. We're keeping them contained. And if you want to have an entire discussion on prescribed fire, we can do that as well. Anyway. Okay, so that's the treatment, one of the treatments. I'm not going to show you mowing. This is what you guys will be doing, this fast. So you can see them, they're very straight on the line because there's a tape stretched in front of them, a transect tape. And so we're putting down a 1 meter square every 5 meters, and then we're identifying everything in there. So that's what it looks like. And this is the layout. So every one of those plots has two permanent transects on them. They're 50 meters long, they're parallel. The one you'll be most interested in is this one down here, where we put the quads. And so it's 50 meters long. Every 5 meters you put down your square. You will be on this side, and then you'll be leaning over and looking into these squares to figure out what those plants are. If you cross to this side, we will yell at you and say, get back. positively hit you with a stick, because what we're trying to do is keep compaction to a minimum in this area, because they're permanent transects, so we go back every year to the same place. So there's a pathway to all of our plots, and we're trying to keep that from happening in the site that we're studying. So the reason we have two, so this is a good scale for herbaceous plants, little grasses and flowers, but it's the wrong scale for trees. And so we have actually a second assessment that we do, where we're looking at just the trees, but generally just the staff does that because it doesn't take a whole crew of people. But the one you will be doing, so we are estimating, as I mentioned, percent cover. So that's the amount of ground covered by a particular species. It's easiest to envision if you think of a live oak with the big drip line. So what you show on a flashlight over that live oak, what is shaded on the ground is the cover, and that's what we're estimating. We're doing it by species, not individual plant. So this is our 1 meter quad. This is plant A, plant B, and plant C. And we are estimating these into categories, coverage categories, visually. And so this will be on the side of your data sheet. It starts very little, so 1%. So the percent that I'm talking about is percent of this square. So the smallest, about yay big. And then 5, so bigger than 1%, but smaller than 5%, up to a quarter, up to half, and then, oh, too far. And then up to 3/4. So we're gonna do a little practice today. Because all the plants will be this easy to identify and separate. It'll be great. Okay, so we're gonna do this as a group. And we're gonna start with this green guy. And so what we're looking for is the total cover of this green guy as collectively. So in your mind, I'm going to test your spatial skills, sort of cluster them all together so that they're just touching, and we're going to figure out which box they fit in. And what helps me, and just what's inside the box. So this part out here we don't care about because it's outside. Mm-hmm. And if there was another one, well, we care about what's growing inside, we care about what's in the box, not outside the box. So even if the plant is rooted out here, if it's got stuff hanging in, you count that. If it's rooted in here but most of it's out, you don't count the piece that's out. We also only look at alive things, and that can get trickier than you'd think. But okay, we're starting with this guy. So what helps me is to think about the breakpoints in these coverage classes instead of trying to go, that's 63.2%. You will argue with yourself forever if you do it that way. So I usually start in the middle. So do we think that this green thing covers more or less than half of this quad? Let's see. Do we think it covers more than half?
I don't.
Yeah.
Do we think it's, a lot of them are right at. So do we think it's less than half? Yeah. Okay, so less than half. Do we think it's more or less than a quarter? More. Okay. So that's we're 26 to 50. So that's our coverage. So let's do this little red guy. So do we think it's more or less than 1%? So see the 1% at the bottom? Is it more than that or less than that? Sorry, is it more? Is it more, or is it more than 5%?
Yes. Yeah.
Yeah. So when you get to that pause, you're getting close. Is it more than 25%? No. Okay. So we're going to put that in probably more than 5, but less than a quarter. So that's class 3. And we don't actually care. You're never arguing over class 6 and class 1. You're arguing over 2 next to each other. The general rule of thumb is you'll be surprised how often they're like right on the line. If you think it's right on the line, then you go up 1 because you're probably missing a few. And we do it as a group because some people estimate high, some people estimate low, and so you sort of come to a consensus in the middle. And there are more sort of precise methods we could be using, but they're a lot slower. And what we're tracking is change over time, big change over time. So we don't actually care if it's 23% or 28%. We care if it goes from 5% to 70%. We want to know those big changes. And this is repeatable and fast enough. It still takes us about 2 to 3 weeks to get through all of our plots, and by that time the season is changing, so we really couldn't do a slower method. This is the other one that you guys probably won't be doing. So this is called a line intercept, and we are measuring the coverage of the trees. It's just they're too big for these little 1 meter quads. And so what we do is we walk the entire, there's 100 meters worth of tape out there, so we walk the entire thing, we measure wherever it's crossing the line, and then you add that up, and that gives you a percentage. Oh, I did precision. Okay. Okay, so, yeah. Yeah, so we usually get the questions like, well, this method seems pretty not precise. And so there's a difference between precision and accuracy. So precision means that all of your measurements agree with each other. Accuracy means that they are reflecting the underlying trend. You can be both precise and accurate. You can also be precise and inaccurate. It's just that your measurements are all, did I do a good one? Yes, like this is precise. It's hitting the bullseye, but it's not, or they're close to each other, but they're not hitting the bullseye. This one is all nicely surrounding the bullseye, but they're not close to each other. This is the goal, but that doesn't always happen. So our method is fairly accurate. It's less precise. But that's about speed and what we can do. And then this is just a plug for the scientific method. Why don't we just do one of these things? So just do one treatment and measure that. So this is why. So you've got two fields. You set that one on fire and you don't set that one on fire, and they're different after that. Great, I have learned something today. But then you're like, oh, the soils are different under there. So it wasn't my treatment, it was because the soils are different. Like, okay, fine, I'll divide them in half. And then I'll do all of these, and then I will know. But then you're like, oh, there's a road up there, so all the north ones are different because there's this other thing that I didn't think about up there. And this can go on forever, especially in ecology. So the way that you deal with that is you do a bunch of copies all over the place, and if all of your copies of one type of treatment are agreeing mostly with each other, then you can be more confident that it's related to your treatment, not some other thing that you didn't categorize. So that's why we got a bunch of treatments of everything. We'd like to have more, but we have limited resources and space. And that's just nature. So are there any questions at this point? Okay. So what we're going to do now is actually go through your books. I didn't give you very long. Any questions, comments, concerns? Let's go through the books. I'm going to grab this one. So these are yours. Take them home, put your name on them. Try to bring them back with you when you come for your survey. We will have extras, but this is what this basically is it's a combination field manual and field guide that we've created. So the other thing that I would do when you go home, there are several sections. I would put sticky notes or something to divide these to make it easier to flip. But the first several pages is just the instructions that you're going to get today. Read that if you want, or you can just listen to us today. The second section, starting on page 15, is a species list, and it's organized by scientific name. So there's a couple of things to look at. So I've given you a book, and I've also given you a sample data sheet. So the species list organized by scientific name on 15 has all your species, it has your common name, and then in the middle here is a code. And this code is what you'll actually be recording. So you can see that that code is created from the scientific name. So it's 4 letters and a number, which is usually 1, but look it up. And look it up in this list is the most accurate, the one that gets fixed most often. You'll start to know these codes, but just double check. And so the reason that we're using these codes is because they're less ambiguous and they are faster to write down than trying to write the entire scientific name. Why don't we just use the common names? I'll tell you, because common names don't mean anything. They're not standard. Every species, especially if it's a common one, has 7 different common names. One common name can apply to a bunch of different plants. Dick and I will call them 4 different things during the same survey, but the scientific names are standardized. They do change sometimes too, and I wish they wouldn't, but they do. But they change a lot slower, and they have been standardized. So we're using these codes so we can get back to these scientific names. And so if you look at your data sheet, there's a section, that middle column, it's where you'll be recording all of the codes. Now, you'll also be recording percent cover, and the percent cover is up there in the corner of the data sheet. We have the cover classes. And that basically leaves one column after you record where you are, which is the quadrat, free. So if you want to record the scientific, if you want to record the common name or the scientific name, you can do that there. which is helpful if we can't read your handwriting or if you get a code wrong. It gives us a second chance. While you're recording these codes, please use capitals because I, L, and 1 all look the same. So think about your data entry person and use capitals for those. Try to write clearly, and then if you give us anything else in the extra side, that's helpful because sometimes we can't. My handwriting is terrible. I am the worst. Now, I'm sure this list is sort of scaring everybody, but so the whole thing is reorganized by common name. And this is where those tabs, our budget doesn't run the tabs, but you all can add your own. Starting on page 25, we've got the common names. So it's the exact same list, just reorganized by common name. And you will probably learn the common names first, but try to start learning the scientific names because then you don't have to have arguments over what plant you're actually talking about. So anyway, they're all there. And as I mentioned earlier, we don't actually expect you guys to know any plants when you come onto the survey. We just need you to be able to see that this plant is different than that plant, and we will help you get to the ID. And we'll probably yell a code at you, but if you don't understand what we're saying, just yell back and say, could I have an actual name, please? And it's good to get in the habit of double checking these things. Okay, so after those two lists, these are our master lists. We have, starting on page 36, it says grasses. So after the sort of business end of this, it turns into basically a mini field guide just for our site. So we have the most common plants that we tend to find organized in here. It's not everything, but it's a lot of them. And it's a lot of the stuff that we see a lot. And the way that these are organized, so if you go to the next page, so this is a very mini field guide. So you'll have a line drawing, you'll have a very, very brief description. You'll also have the code and the name again up here, but these ones, whenever we change something in the main species list, it doesn't always propagate all the way through. So if you find a mistake, let us know, but double check those names in the master list. If you've never seen a plant before, you're not going to get to it from this description. It's too brief. The goal here is that we will help you learn, and you take notes in here. You can tape samples, not from inside the quad, but from outside the quad. Tape samples in here, take your notes, and then you'll have a good record. So when you're learning your plants, especially when there's a bunch of them, your notes, like your first impression, you're going to have that same first impression every time. So write it down on here. It's not going to be what's in the keys, but you'll remember, oh, right, that's the fuzzy one with the white line on it. Because you'll come and you'll see it again, you're like, well, what's the fuzzy one with the white line? And if you've written it down, you can get back to it. So write those things down, and the samples are very useful, having those. And then this is also why I want you to write your name on it.
Did you tell them to write their names?
I did,
Okay.
but I did again. So behind us we have Dick Davis, who is our master tape stretcher and identifier.
They generally try to keep me away from the volunteers as much as possible.
We have a hierarchy of who can identify plants. I'm your first brush, and then Dick's your second one, and then there's someone named Manette who is third, who's usually in the lab that we can't get it. we bring her stuff. I know she's retiring, but we're totally sending her stuff. We can't not be here. Okay, anyway, so we have a grass section. And then we do the same thing with forbs. So what are forbs? Remember what a forb is? Yeah. Forbs are your flowers. A lot of people think of them as the weeds, whatever, but they're not grasses and they're not trees. We got a forb section and then we got a woody plant section. So the vines fall into the woody plants. And again, this is not comprehensive, but it's a lot of what you'll see out there.
Are there invasive and native species?
Yes. Yeah, so this is all the things that we tend to see. And then the last section, we have a list of field guides that we liked in 2012 and continue to like. Someday we'll update this. That is the... All right, so any questions at this point?
What are the ones in bold?
We need to take this out. They're a bit more common, but I think next year we're taking the bolds out.
Yeah.
Because I think it's skewing things. We do have, because we have species listed in here that we've never actually found. We just kind of think they might be in here someday. I think that's the reason the original bolds went in, but just ignore that. And then we're going to head out into the field and do some practice. And so the way you read these tapes, first of all, if the numbers look upright to you, correct, then you're on the right side. If they're upside down, you're on the wrong side, and you have to jump back to the right side before Dick or me notices and hits you with something. You don't want to squish stuff over there. So the red numbers are the meter numbers. So you're looking for, I'm right in the middle of one.
29.
There's 29, let's find 30. 30, here's 30. So we're gonna be putting quads down every 5 meters. So you're looking for some multiple of 5, some red multiple of 5. And then the smaller ones are the portions of that meter. So 30.1, 0.2, 0.3. None of that matters to you except to find this multiple of 5 or 0. We put them at 0 and then we put them all the way up to 45. Again, we just covered being on this side. What we're gonna do out here is do one practice one all together in a big mass. You won't be able to see very well, but you'll get used to the process. And then we're gonna break up into smaller groups and use these quads that we're setting out. Just give you some practice. Looking at these things, estimating coverages. And again, you don't need to learn any plans today. If you learn a couple, that's great. But the point here is to learn how to do this. So.
Should we always be confirming with you what the species is?
Yeah, if you're at all confused, and even if you're not, we like to walk through and we like to just kind of make sure everything is what you think it is, or what you think it is. Sometimes we're wrong and you can argue with us. But just for quality purposes, we're not going to check every single thing that you do, but we're going to poke through your quads to make sure that we're mostly in agreement. Page 12. Alright, so once you're at page 12, there's 2 things showing here. One is the herbaceous data sheet. So you've already seen a version of this. This is what you'll actually be recording on. And then next to it is an unknown data sheet. So sometimes it happens, we don't know what a plant is and we can't figure it out. So in that case, we would take a sample of it and take it back and identify it later with all the books and microscopes if we can. So that's what happens. Y'all are generally not going to be taking samples of unknowns. If there's something we can't figure it out, we'll take the sample. We're trying to find one that's not in the quad, ideally. But that's just something that's good to know. So this is your herbaceous data sheet. There's a couple things to notice. At the top, it says plot number. So when we arrive at wherever we're studying, we'll yell out the plot number. And so that's the same for, like, this entire line would have the same plot number. So you record that once. It'll be some number between 2 and 54. And make sure you got that written down because it tells us where you are. The next one is the researchers. You can just record the first name of whoever's in your group. And then the date. The date's important. We want to know when this data got taken. So record all of that at the top before you start anything. And then the next thing to record, and this often gets forgotten, is the quad number. So this first column here, quad number. So that's how far down the tape you are. That's that red number that's some multiple of 5. Today we may not be at multiples of 5, but on the real starts.
Yeah.
Oh. So good. Okay, it will be. It should be. If you're not on a multiple of 5, you're in the wrong spot. So you need to find one. Make sure that gets recorded because then if we don't know where you were. Once you recorded that, this column has no other purpose. So this is where people like to often just record common names or notes or something. Don't record too many notes because I'm taking this from you at the end of every transect. So put your notes in your notebook. But if you want to record the common name or even the scientific name, it helps people learn.
It helps you learn. It helps us interpret your horrible handwriting.
Yeah. But this is not the official recording there. The middle section where it says species is where you're going to record those codes that we looked at earlier. And then there's 2 things at the top, bare and lit. So bare ground, we're going to be recording how much bare ground there is and then how much litter. And by litter, what I'm talking about is plant litter. So this stuff, if there's a Coke can or whatever, we'll pick that up. It's not part of the research.
Dead plant material that's no longer attached to the parent material.
Yeah, which is what we'd say. Okay, and then, so what we'll do when we get to the quad is you'll record all this stuff, then you'll get on your knees, or maybe stand up at first, record bare and litter, and then you'll record all of the species, and you won't worry about cover yet. Just record everything you see. And there's a couple of reasons that we do it that way. We don't want you to do the percent cover until the very end. First, because if you try to do species and then cover immediately, you're going to miss stuff. But then also,
you'll get so hung up on arguing about the percentage of cover, you'll forget to write down some of the species that are in there.
Yeah, you'll miss species, and you also have a much better idea about the cover after you've dug through the whole plot, because there's always a bunch of little stuff that's underneath things, and you just have a better sense of that after digging. So, record species, then get back, and I usually stand up for this, but you can do it anywhere you want, and then you do the cover. And again, the covers are up here. I may not have mentioned this. Most of the codes have a number. They all have a number. It's mostly 1, but occasionally it's 2 or 3. And that happens when the 4-letter code has a duplicate. And so the number will designate a different plant. So check these things. Don't always assume that it's 1. There's a couple of common ones that have 2s instead of 1s.
But all the ones that are not 1s are listed on the edge of your data sheet. So.
They are, but still go back to the master list. And the goal here is not speed. Like, take the time at the end of the plot, check those codes, get them right. You will learn them and you will start going faster, but we're not trying to get through fast, we're trying to get through correctly. So, plus it's kind of nice when you're done with the plot to just take that little bit of time to double check. Sit down in the shade and check your numbers. Yeah, bare and lit. Okay, so bare ground is rocks and it's bare mineral soil. So this stuff is not bare. If I had all this litter gone and I could see the actual dirt, that's bare. So in this one, oh, and we should look at the dividing line. So most of these have marks. So these two little marks right there, about the size of a softball, that's your 1%. And then this is the 5%. I don't have a good, so 5% is about yay big. A quarter is a little easier to visualize, so that's here. And then half, and then 3/4, 75%. Those are your break points. I feel like the littlest ones are the hardest to kind of to visualize. So we're gonna start with bare ground. So I have rock. This rock is bare ground. Everything else, all this is covered in litter, or it's covered in a plant. So if a plant is hanging over and you can't see the bare ground standing up, I'll get to the litter in a minute.
I mean, we ignore the trees hanging over.
Yeah, we also ignore the trees.
For this part.
So for this part, when you're trying to figure out bare ground and litter, ignore the trees. I'll look down here. So, but if you can't see the bare ground standing up, it's not bare. It's covered in a plant.
Do we ignore shrubs too?
Yeah. So
For this part.
above this sort of bottom layer, we're ignoring that.
So.
This is basically, we're looking at the rocks for bare ground right now. So is this, I'm gonna start at the bottom, is this more or less than 1%? Is that bigger than a softball? More, thank you. More or less than 5%? More or less than a quarter? Getting close. Looking down at it, I probably go over. But it's close, and so you just listen for that pause in your group. More or less than half. Less than half, so it's probably in the 26 to 50. There's maybe an argument to be made for the smaller one, but I think it's over that. So 26 to 50.
Some people always estimate high, some people low, and it just kind of evens out, over the 20-something years we've been doing this.
Yeah. So to record that, there's a number next to those cover classes. So 1 through 5 or 6. That's what you would record, is that number in the final column.
So feel free to debate and disagree.
That's
part of the process, and good. So
It's
don't just let
encouraged.
other people,
If 3 people say
lead
class
it.
3, and one person says class 1, it's a class 3, unless the person that disagrees is the person writing it down.
Yeah, the scribe has a disproportionate amount of power. So you're like, oh yeah, you're totally right. But generally,
Don't
the
do
disagreements are not. Anyway, you're usually arguing between 2 that are next to each other. And so again, rule of thumb, go up 1 if it's right on the line.
Average up.
Yeah.
Especially with the plants, because a lot of times there's little baby plants that you can't really see, hidden. So.
Yeah. If there's a complete knockdown, drag out, we will be the arbiters, but hopefully y'all can just work it out amongst yourselves.
Okay, litter.
Litter. Okay, litter is different. So you do bare ground standing up. Don't touch anything. Just look. For litter, though, you have to touch things. So you got to get up underneath these plants, because if I pull these back, the litter is covering all of the ground.
Right up to the base of the plant.
Yeah.
So if you we've done this. If you clipped off all the plants within the square, then all the tall stuff, then you're looking at the ground surface. Everything in there would be either bare ground, litter or the base of the plants. The base of the
Base
plant.
of the plant is important. The plant has a spread.
Yeah, and so estimating the litter, you want to count the litter that's all the way up to the edge of this clump of grass. So you got to kind of poke around. It's the litter estimation is in a lot of ways the hardest thing to estimate.
But, okay, we're gonna do the same thing again, and I realize you guys are not very close. But we'll do it again with this. So, more or less than 1%. More or less than 5. More or less than a quarter. More or less than half. Yeah, I think it's gonna be, is it more or less than 3/4?
If you're going base of plants, this is a clarifying question real
Yeah.
quick. Okay, so if we're expecting bare and lit to almost get to 100%, seeing that that's like the whole
Bare,
bottom of
lit.
the, like with the plants being only the base of the plant being maybe 10 to 20%. Are there 200%, or at the end
You're
of
gonna
our
end
little
up-
quad, does everything add up?
It's gonna add up to way over 100.
Okay,
Because
great.
this tree is covering most of the plot. These things are covering, so they crisscross.
Okay,
So we're
good.
actually not gonna get, and plus, they're cover classes.
Yeah.
We could get it to 100 if we cut all these off and measured each base.
Right.
This is the only thing that'll get.